Have you ever wondered how white blood cells attach to bacteria? How cytotoxic leukocytes actually kill bacteria? Watch this video to find out the answers. Welcome to Dr. Exam Questions and Answers. Our questions and answers are based on the deep pathophysiology of acute inflammation. Before we start, we encourage you to support our channel. How? By your likes, comments, share, and subscribe to this channel. What are the main events leading to transmigration of leukocytes across the vessel wall? The key events in leukocyte transmigration across the vessel wall are margination, rolling, adhesion, and transmigration. The first one is margination. Slowed blood flow allows neutrophils to move closer to the vessel wall. Rolling, neutrophils roll along the endothelium mediated by selectin interactions, establishing loose contact. Adhesion, activation of leukocytes and endothelial cells lead to the expression of integrins, which firmly bind leukocytes to the endothelium. Integrins function by binding to the intercellular adhesion molecules and extracellular matrix components like fibronectin and laminin, all together so as to provide a strong and stable adhesion. Role of selectin in extravasation is to promote the movement of leukocytes toward areas of inflammation. So they take part in both the margination and the rolling phases. Now for integrins, their crucial role is in adhesion and transmigration phases because they facilitate firm binding of leukocytes to the endothelium, enabling them to exit the bloodstream. The next question: how are leukocytes activated? Leukocyte activation is primarily initiated through the engagement of surface receptors on their plasma membrane. So the initiation process involves binding to the endothelial cells or interleukin receptors, then signal transduction process. Here there is activation of phospholipase C that will lead to the production of diacylglycerol and inositol triphosphate. The last process is calcium elevation. Yes, increase the level of calcium in the cytoplasm will enable intracellular processes for leukocyte action. The next question: what are the signs of leukocyte activation? Leukocyte activation is characterized by number 1, expression of adhesion molecules, number 2, cytoskeletal changes, degranulation, oxidative burst, and the last one is protein synthesis. The next question: what is chemotaxis? Well, the answer: chemotaxis refers to the directed movement of cells in response to a chemical gradient created by a chemoattractant. This process involves active navigation towards areas of higher concentration of the attracting substance enabling cells to migrate effectively toward signals linked with injury, infection, or inflammation. What are the most important chemotactic substances? The two main types of chemotactic substances are exogenous and endogenous chemoattractants. Exogenous chemoattractants are derived from external sources, particularly bacterial polypeptides. These chemotactic substances attract immune cells, attract leukocytes toward the site of infection, the site where there is infection. Now, endogenous chemoattractants are produced internally, are produced within the body, particularly from stressed cells, from the cells which are damaged. Example, we have the product of complement system activation, such as C5a, which plays a crucial role in amplifying the immune response. We also have arachidonic acid derivatives, leukotriene. These are generated through the lipoxygenase pathway, and they are known for its potent chemotactic effects. Cytokines. They direct neutrophil migration and also activation. The example of cytokines, we have interleukin 8. These are also among the endogenous chemoattractants. How do chemoattractants act on leukocytes? Well, chemoattractants exert their effect on leukocytes through a series of activation steps, which include binding, G protein activation, phospholipase activation, calcium mobilization, and microfilament assembly. So the chemoattractant substances first bind to receptors on leukocytes on white blood cell membrane, initiating a signaling cascade. This binding now triggers the release of G proteins, which are crucial mediators in cell signaling pathways. Now, the activated G proteins stimulate phospholipases, leading to the production of the secondary messengers. We have DAG, the diacylglycerol, and inositol triphosphates. Pseudopods derive from the Greek term meaning phosphates. They are just extensions of the cytoplasm that emerge from the leading edge of activated leukocytes. The structure and composition. These cellular extensions are primarily composed of aggregate of microfilaments, which are made up of both actin and myosin. The mechanism of action of pseudopods. So myosin contraction. The contraction of myosin fibers lead to the shortening of the microfilaments. Now this contraction effectively pulls the rest of the cytoplasm towards the tip of the pseudopods, facilitating the movement of white blood cells. Pseudopods play a crucial role in the motility of leukocytes. Remember, leukocytes should move toward the site of infection, and the movement is facilitated by pseudopods. How do leukocytes kill bacteria? Well, leukocytes employ a three-stage process so as to eliminate bacteria, which is crucial for the body's defense against infection. The The first one is attachment. The initial stage involves the attachment of bacteria to the surface of the white blood cell or the leukocyte. Number 2 is phagocytosis. The bacteria are then engulfed by the leukocyte through a process called phagocytosis, in which they are taken into phagocytic vacuoles where they're going to be destroyed. The third stage is killing and degradation. The final stage involves the destruction and breakdown of the engulfed bacteria. The key step in killing bacteria is phagocytosis. This involves opsonization. Opsonins, which are molecules that mark the bacteria for destruction, cover the surface of the bacterium, making it easier for the white blood cell to recognize that this is a bacteria and then to bind to it. Receptor binding. The opsonized bacterium binds to receptors which are found on the surface of the plasma membrane of the white blood cell. Invagination. The plasma membrane invaginates, forming a phagocytic vacuole around the bacterium. Intracellular digestion. The phagocytic vacuole fuses with the lysosomes, leading to the release of digestive enzymes that break down the bacterium into smaller components. So yes, the key step in bacterial killing is phagocytosis. How do leukocytes attach to bacteria? Well, leukocytes attach to bacteria through a non-specific process where bacteria initially bind to the leukocyte surface. Now, to enhance this attachment, body fluids coat the bacteria with opsonins, which are proteins that facilitate recognition. Leukocytes possess specific receptors for opsonins, enabling more efficient and effective attachment to opsonized bacteria compared to other particles. What are the main opsonins? The answer? Immunoglobulin G, IgG, a key antibody involved in immune responses that enhances phagocytosis. The complement 3b, C3b fragment. This component is generated during the activation of the complement cascade and plays a crucial role in marking pathogens for destruction. Then we have colectins. These are carbohydrate binding proteins such as mannose binding protein that function as natural lectins, binding to specific sugars on pathogens and then facilitates their recognition by immune cells by white blood cells. The next question, how do leukocytes form phagocytic vacuoles? Leukocytes form phagocytic vacuoles through the invagination of the cell membrane and elongation of cytoplasmic processes. This process depends on cytoskeletal restructuring activated by metabolic events similar to those in leukocytes responding to chemo-attractant substances. What are the main bactericidal substances used by neutrophils Well, bactericidal substances are substances that kill bacteria. Now, what are those substances? Number one, we have hypochlorous acid in oxygen-dependent killing. We also have number two, lysozyme, lactoferrin, defensins, and acidic environment in oxygen-independent killing. What two mechanisms are involved in bactericidal activity of neutrophils? Like mentioned, we have oxygen-dependent killing and oxygen-independent killing. Now, in oxygen-dependent killing, which relies on the oxidative burst and generates reactive oxygen species. Then, in independent oxygen killing, this relies on antimicrobial substances like lysozyme, lactoferrin, and cationic proteins. What are the key steps involved in oxygen-dependent killing? Well, the key steps are, number one, generation of a superoxide from NADPH oxidase. Number two, conversion of superoxide into hydrogen peroxide, H2O2. Number three, reaction between hydrogen peroxide and chloride ion so as to produce hypochlorous acid, catalyzed by the enzyme known as myeloperoxidase. What are the clinical consequences of congenital defect of abnormal leukocyte functions? Well, individuals who have that defect in leukocyte functions, including phagocytosis and bacterial killing, they face a significant increased risk of either recurrent or severe infections. These individuals, mostly infants, they are susceptible to opportunistic infections, which are coming from non-pathogenic microorganisms, and their immune systems cannot fight against these threats. The consequence, severe infection will come about, and also morbidity and potential mortality can come because of that defect in the functions of white blood cells. Additionally, chronic exposure to repeated infections can result in lasting health issues like lung damage from recurrent pneumonia. Furthermore, some congenital defects may also predispose individuals to autoimmune diseases because of that imbalance in immune responses.